Welcome, everybody, to this edition of the Connect Science virtual seminar series. I want to bring your attention to the website that you see on the slides here, the Connect Science website. Please visit there for both past recorded sessions as well as this one, which will be recorded, and future as well. Today, I'm very happy to introduce Scarlett Lynn-Gomez, who is the professor at the Helen Diller Comprehensive Cancer Center and the Department of Epidemiology and Biostatistics at the University of California in San Francisco. She's also the co-leader of the Cancer Control Program at the Helen Diller Family Cancer Center, and as well the PI and director of the Greater Bay Area Cancer Registry. So she's got a lot of interesting things to say and great thoughts, and will present today about multi-level studies to address cancer health disparities from neighborhoods to omics. And let me just also mention before Scarlett starts that we will have questions, so type your questions in the Q&A and we'll answer them at the end of her presentation. Thank you.
Great. Yeah. Thank you, Tim. Thank you for that nice introduction, and thank you for inviting me here. It's a pleasure to be able to share with this group just some of the work that my group at UCSF is doing on multi-level studies, and I'll give you a few examples of how we're trying to integrate data ranging from neighborhoods to omics and putting it all together for addressing cancer health disparities and inequities. So I like this quote from Paula Braverman. It's a bit old now, from Public Health Reports in 2014, but I think there's no really better quote that really sets the stage and the context for why we do the work that we do. So she speaks about what health equity means, that health equity means social justice in health. That is, no one is denied the possibility to be healthy simply because they belong to a group that has historically been economically or socially disadvantaged. We use this framework to help to guide the research that we do. This is the Cells to Society model that was developed by the Centers for Population Health and Health Disparities, a program that was funded through the NIH, several institutes within the NIH. And what this group of investigators did was to develop this framework for conducting multi-level research. There's been several various similar kinds of multi-level models. Tim himself has developed one really important model as well. So this is not necessarily to say that this is the one and only model, but for us, it fits the work that we do quite well. And in particular, what we like is that the different factors at different levels as conceptualized within this model are actually explicitly ordered. So from the bottom here, let's start from the bottom, we have the biologic factors ranging from biologic and genetic pathways, such as biologic processes, genetic mechanisms, to responses at the biological level, depression, stress, hypertension, to individual level factors, including risk behaviors and individual level demographics, social and physical context, including the physical context, social relationships and social context and environments. And then here at the top, we have institutional context and policies. And so I want to draw your attention to these factors at the top, which are the fundamental causes as proposed by the Cells to Society model. This is defined as having roots embedded in policy, shared social norms about health and social practices, socioeconomic disadvantage, and policies that affect availability of services, including who receives them and the level and quality of the service that received. And the idea with this model is that as we go up from the bottom to the top, we get the relative influence or the impact on disparate health outcomes is more important. So that is to say, these factors at the top are the most important in terms of driving health disparities and health outcomes. And I want to draw your attention also to the arrow at the left, which emphasizes the importance of the interactions across these different levels and these different factors. Now, as we sort of move downstream, we come to the social determinants. These are social determinants of health, the conditions in which people are born, grow, work, live, and age, and the wider set of forces and systems shaping the conditions of daily life. This is a common definition used by the World Health Organization. And so these include the social environment, including the degree of collective efficacy or social capital, racial ethnic integration within the neighborhoods in which we live, the relationships that we have. That research shows that has been shown to shape our health behaviors, the physical and built environment, including the quality of the neighborhoods that we live in, the man-made aspects of the environments that we live in, and the individual level social-demographic characteristics of people living in these environments. So now I want to share with you how just two studies, two research programs of studies in which we've tried to apply this multi-level cells to society model to persisting health disparities. So the first of this is looking at lung cancer among Asian American females who've never smoked. I'm going to just try to. Sorry, just, okay. I had the little WebEx panel and it was obscuring my view. Why do we care about lung cancer among Asian American, Native Hawaiian, Pacific Islander females? We know that they're an incredibly diverse population. In fact, the AA-NHPI population comprised more than 30 people from 30 different countries speaking more than 100 different languages. We also know from registry data that lung cancer incidence varies greatly across the different AA-NHPI groups, and from case series studies, such as a group of lung cancer cases diagnosed within one particular healthcare system, for example, we know that more than 50% of lung cancer cases among AA-NHPI females are never smokers. And this is quite high in contrast to what we see among the general US female population of about 15% never smokers. And until now, we've had no sufficiently large data source to be able to document what is the incidence of lung cancer by detailed AA-NHPI race, ethnicity, and by smoking status. And this is because we don't collect smoking data in cancer registry data. So to address this question, this major gap, Dr. Iona Chang, my colleague, and I proposed and were funded on a study funded through the National Cancer Institute, in which our objectives were to firstly assemble a multi-level integrated data set relying on existing data. With this data set, we conducted incidence analysis by smoking status and detailed race ethnic groups. And we also did this by lung cancer histology and by EGFR and ALK mutation status. That was our original goal. And then focusing specifically on the Asian American, Native Hawaiian, Pacific Islander female never smokers, we used this integrated data set to assess risk associated with 5 different buckets of risk factors: reproductive factors, body size, previous lung diseases, infections, and air pollution measures, as well as social environment measures. Here's kind of a flow chart of how we assembled this integrated data set. So we started with electronic health records data. That's the EHR data boxes that you see here. And actually at the time that we wrote the grant, we explored collaborations with several different EHR healthcare systems that had robust EHR systems. We ended up collaborating with 2 systems, Sutter Health in Northern California and Kaiser Permanente, Hawaii. Sutter Health in Northern California is actually the 2nd largest healthcare system in Northern California, second next to Kaiser. It covers about 30% of the Northern California population and has a very large population of Asian Americans within their membership. It's a multi-payer healthcare system, so it's a mixed payer, so it's not an integrated healthcare system like Kaiser. The other healthcare system that we collaborated with was Kaiser Permanente Hawaii, which obviously is a much smaller healthcare system that has a very large population of Asian Americans, Native Hawaiians, and Pacific Islanders. So we extracted the data on all members based on our eligibility criteria, who ultimately went on to develop lung cancer, but also those without lung cancer, pooled the data together and ended up with the cohort of over 2.2 million people, of which 1.275 million were females. We then extracted and harmonized data from these two EHR systems. And this box here shows you the data that we pulled. Unfortunately, even though one of our original aims was to extract data by EGFR and ALK mutation status, through a lot of research, we learned that those data were actually very difficult to extract without a lot of work from these two EHR systems. So that's work that we're going to be looking into, perhaps using approaches like natural language processing to extract. We then took all these 2.2 million individuals within this cohort that we defined and geocoded all of their addresses. We actually geocoded 2 points of addresses, one at a point in time that we defined as cohort entry, and then one at a later point in time. Once we've geocoded the addresses, we can actually now link in all sorts of different geospatial data. So for this particular study, this grant, we proposed to look at a measure of neighborhood socioeconomic status and measures of ethnic enclaves, as well as air pollution measures, in particular, particulate matter exposures, PM 2.5 and PM 10, and data on traffic density. We then linked this cohort to the cancer registry data. And that was how we were able to basically virtually follow up the cohort over time for incident lung cancer. We identified over this follow-up period, we identified over 7,000 lung cancer cases in Hawaii and California, of which 3,867 occurred among females. And then we extracted various tumor factors from the cancer registries. So here's some descriptive data from this study, and this has actually just been recently published in the journal Cancer Epidemiology Biomarkers Prevention, and we have a follow-up paper that's in a revise and resubmit mode. So this is just from the females who were diagnosed with lung cancer, the 3867 cases. And what I'm showing you here is the distribution of smoking status among these lung cancer cases. The blue are the never smokers. The first thing you'll notice is that there is a fair percentage of cases with missing smoking status. We did actually a lot of work to try to figure out how to maximize information on smoking status from their EHR systems. And in fact, these two healthcare systems do an outstanding job of collecting data on both race and smoking status relative to other healthcare systems that we had explored. And then, so what I'm showing you here are the distributions by detailed Asian American, Native Hawaiian, Pacific Islander race ethnicity, as well as for non-Hispanic white female cases, Black or African American female cases, and Hispanic or Latinx female lung cancer cases. So just kind of focusing your attention on the percentages that I've highlighted in red here. Let's start here at the bottom. So among non-Hispanic white women, females diagnosed with lung cancer, 21% were never smokers, 14% among Black females with lung cancer, and 38% among Hispanic females with lung cancer. In the AA-NHPI population overall, we saw 38% never smokers. Looking just among Asians, Asian Americans, we saw 50% never smokers. But there's already, you can see here just from this chart, tremendous heterogeneity across the AA-NHPI groups, whereas we see 14% never smokers among the Native Hawaiian females diagnosed with lung cancer, compared to 79% among Chinese females diagnosed with lung cancer. So I want to just kind of pause here and reflect on this number, the fact that nearly 80% of Chinese females who were diagnosed with lung cancer have no personal history of smoking, which is incredibly, incredibly high. And this is the first time we've seen these data broken down by a detailed Asian American ethnic group. And now, because of how we designed the study as a longitudinal cohort study using the EHR data, we were now able to calculate incidence rates. So, and this is for the first time we've had data on incidence rates by smoking status for detailed Asian American ethnic groups. And so what you can see here is these are incidence rates of never smoking lung cancer, specifically among females, age adjusted. Let's go down here for non-Hispanic whites. The rate is 10.1 per 100,000. For Hispanics, it's quite similar. The confidence interval is a bit wider. And unfortunately, we didn't have enough numbers to be able to calculate a stable rate for Black females. But what's interesting here that I've highlighted in the red box is that for nearly every Native Hawaiian, Pacific Islander, and Asian American group, the incidence rate of lung cancer is almost 1.5 to 2 times higher, 2 times higher for Asian Americans and about 1.5 times higher for Native Hawaiian, Pacific Islanders when we compare it to the non-Hispanic white female group. And for the most part, we also calculated relative risk ratios where we took the ratio between these rates compared to non-Hispanic whites. We've also compared it to Asian Americans overall. We've also compared it to everybody combined in the denominator. And we find that the differences are statistically significant. The interesting exception here is for Japanese women, where we see that actually their rate of lung cancer among never smokers is quite low, in fact, lower than what we see for non-Hispanic white females, although of course you could see the confidence interval is very, very wide. So we're wrapping up the study. We are in the process of concluding our risk analysis, where we're looking at risk associated with these different exposure domains. These first 5 we extracted from the EHR. These, again, were based on geocoding and geospatial data that we've compiled. And we also have an administrative supplement. This is also work that we're wrapping up, led by my colleague, Mindy Dirawan, where she is applying a machine learning approach called iterative random forest to look at the joint contributions of the multiple multi-level factors to lung cancer risk. We were able, because that study entirely focused on existing data within EHR systems, excuse me just for a second here. Oops. Sienna, there's things in the freezer at the very top, acai bowl. Sorry about that. My daughter was looking for her breakfast. We then were able to propose a follow-up study, and we're funded on a second study where we have now the opportunity to collect de novo data to be able to address this question further. So this is a fairly newly funded R1 from the National Institute of Minority Health and Health Disparities. My multi-PIs, Dr. Moon Chen from UC Davis and Dr. Iona Chang from UCSF are leading the study together. And we call this the FAN study. FAN stands for Female Asian Never Smokers. And we have 2 broad aims. First is to identify the attributable risk of known putative and suspected multi-level risk factors, particularly focusing on factors accumulated over the life course for lung cancer risk among Asian American female never smokers. Specifically, we propose to focus on the independent effects, but also the interactions between genetic ancestry, individual level exposures, and especially here focusing on those that may be more culturally relevant in these population groups. And then also contextual level risk factors, including indoor and ambient air pollutions and social environmental factors. Our second aim is to characterize the mutational landscape and identify associated risk factors, the multi-level risk factors from the first aim that may be associated with these different mutational landscapes that may be unique to the Asian American females with lung cancer. In terms of our study design, we designed this as a population-based case control study. We will be identifying and recruiting, our target is about 600 cases from our Greater Bay Area Cancer Registry, and we're using a method called early case ascertainment, which relies primarily on electronic pathology records that come into our registry in an almost real-time basis. And so we've set up programs to be able to screen these electronic pathology data for potential eligible cases as they come in. More than half of these we expect, just based on the distribution that we see, are among Chinese women. About 110 Filipino women, 90 Vietnamese, and then 85 of other Asians of other racial ethnic groups. And then we hope to recruit and enroll about this comparable number of matched controls. And this is where we've developed an approach to really, we know in terms of population-based case control studies that any single approach you use, whether you're using random digit dialing or neighborhood block walking or using a directory, every single one of those is biased. And this is in large part probably the biggest reason why case control studies really have gone out of favor, because we realized, we've come to realize the significant amount of bias associated with controls. But we had developed an approach where we try to balance out that bias associated with any single approach by using multiple approaches, but in a very targeted way. And we've been able to do this for Asian Americans within the Bay Area. So some of the approaches we're going to be using are recruiting via directories and volunteer registries, lots of community outreach, working with healthcare centers, federally qualified healthcare centers, community organizations, religious places of worship, using various online approaches and ethnic media. And then our second aim will be using whole exome sequencing. This is going to be done by our colleague Cliff Tepper from UC Davis to characterize the mutational landscape of tumor samples that we will be obtaining from the tumor resections. So we're in the process of recruitment, and hopefully within a year, we'll be able to start to report out some preliminary results. So now the second study that I want to share with you, in which we try to apply this multi-level health equity approach, is focusing on prostate cancer, and specifically prostate cancer among African American men. This is well established. This has been a long-standing disparity of higher burden of prostate cancer among African American men. And these statistics I've been updating these for the past 5 years, they've barely budged. This is based on the latest cancer facts and figures that show that the incidence rate of prostate cancer is 80% higher, comparing African American to non-Hispanic men, and the mortality is over 2 times higher. But moreover, we also know that African Americans are more likely to be diagnosed with more aggressive tumors when they are diagnosed. But we also know from studies based on VA data as well as clinical trials data where that suggests that once we're able to adjust for stage of diagnosis and equalize the treatments that are received, we may actually see comparable survival. And in some cases, a few studies actually show even better survival comparing African American to non-Hispanic white men. But population-based registry data show, even after adjusting for these factors, a continuing disparity. So with that in mind, we developed a conceptual framework in which we hypothesized that really race, that is African American race, is really racism. And as a part of racism, there's embodiment of stress that comes from social stressors occurring at multiple levels and over a life course. So this is kind of a framework that we use to think about how might we start to take a different approach than what has been used before to address this persistent higher burden of prostate cancer. So this is the schematic here illustrates the specific kind of components that we propose to look at. So the life course social stressors we hypothesize occur at, again, multiple levels, specifically the contextual level. At the contextual level, we have exposures to structural racism, which imposes racial residential segregation and differences in social environments and built environments. At the individual level, experiences with discrimination, both major discrimination as well as day-to-day or everyday discrimination, adverse childhood experiences, and adverse experiences adversity experienced over the life course. These in turn, especially over time, produce stress, which is embodied and impacts biology and our immune systems and our ability to ward off disease and ultimately prostate cancer aggressiveness and mortality. We also hypothesize that this pathway may be moderated or there may be effect modification by prostate cancer risk loci and or levels and or genetic ancestry. And in fact, there's some suggestion of contextual level social factors and its impact on prostate cancer biology. And so in fact, these first two studies were early studies by my host, Dr. Rebeck and his colleagues, first showing that social adversity is associated with shorter time to disease progression. And they found a significant interaction between selected genotypes and neighborhood factors. And this was data from the Pennsylvania Cancer Registry. Also using cancer registry data, they showed socioeconomic deprivation is associated with Gleason score. And our group more recently showed that neighborhood socioeconomic status in conjunction with individual level, educational level, is associated with both prostate cancer risk and prostate cancer survival. And in fact, for the survival study, we found that these factors, but in particular, neighborhood socioeconomic status, slightly explain the survival disparity between Black and white men. And we also have some evidence from basic science or biology studies demonstrating a potential biological mechanism for stress embodiment in prostate cancer etiology or carcinogenesis. These studies collectively show that stress exposure, these are in vivo and in vitro studies that show that stress exposure can increase the development of prostate tumors, can decrease the effectiveness of androgen deprivation or hormone therapy, can result in less programmed cell death and less clearance of tumor cells, and can produce neuroendocrine signaling effects on the tumor biology process, growth, metastasis, angiogenesis, and apoptosis, and this seems to frequently occur through an immunological mechanism. So sort of putting all of this together, collaborators and I proposed a PO1 grant to the National Cancer Institute called the RESPOND study, which is addressing the role of social stressors, genetics, tumor genomics, and biology. RESPOND is an integrative set of studies that's all focused on this common theme of identifying the multi-level determinants and characteristics of aggressive prostate cancer among African-American men. This was ultimately converted to a U19 mechanism through the NCI, so it's a collaborative effort with NCI. The overall PI of the study is Dr. Chris Heyman from USC, and we have these four distinct studies or projects. The first project, which I will be focusing on in the next few slides, is led by myself and my team at UCSF. The second project is looking at prostate cancer risk loci and genetic ancestry, and that's led by Chris Heyman at USC. Third project is looking at tumor factors, tumor genomics, and this led by Dr. John Carpton and Dr. Franklin Huang. And the fourth project is looking at tumor immune biomarkers, and that's being led by Karen Spanos. This is a national study and so these are the sites that you see here on this map. We're recruiting from California, Texas, Louisiana, Georgia, Detroit, New Jersey, and we've recently expanded to North Carolina as well, and Florida. The recruitment is occurring primarily through these population-based cancer registries, although we also take volunteers as they come, as they're interested in volunteering for the study and if they're eligible. So overlaying these projects onto that conceptual framework, Project One, which is the one that my group at UCSF is leading, is focusing on the life course social stressors, and these are the measures that we are operationalizing and measuring. Project 2, which is led by Dr. Chris Heyman, is focusing on the moderating effects of prostate cancer risk loci and genetic ancestry. Project 3 is focusing on tumor somatic mutational profiles, and Project 4 is the tumor inflammation biomarkers. And our goal is to assess ultimately, after all of this data is collected, our goal is to collect this information from among 10,000 African-American men diagnosed with prostate cancer, again, primarily identified through these state-based cancer registry programs, and look to see how these factors in these different boxes interact to impact prostate cancer aggressiveness among this cohort of men. We are administering a survey to the men who are recruited into the RESPOND study. And within the survey is really designed to capture individual level data, specifically social stressor and other potential confounding information. And again, we're trying to apply this life course multi-level framework. And so the social stressor data we are collecting include self-reported information about their perceived neighborhood environment over the life course, experiences with discrimination over the life course, as well as day-to-day, but at different periods and in time, medical mistrust, adverse childhood experiences, and a form of active coping called the John Henryism. We're collecting a number of different information on their social-demographic characteristics, as well as family personal health history, prostate cancer treatment, and aspects of their diagnosis, and lifestyle and health behavior factors. And then through geocoding their address at the time of diagnosis, as well as the residential history, we are collecting information used from existing geospatial data to operationalize these major contextual factors that are listed here. Our main factor of interest is structural racism. For these measures, we're working with our colleague, Kirsten Beyer from Medical College of Wisconsin, who has conducted really innovative work using data from the Home Mortgage Disclosure Act to develop a measure of redlining and an index of racial bias in mortgage lending. And I will show you in a little bit some preliminary data we have on how these measures distribute across our cohorts. We are also including the standard Massey-Denton measures of racial ethnic segregation at the metropolitan statistical area level, as well as more localized measures of racial ethnic composition, including % Black, and also a measure of typologies, different combinations of different racial ethnic groups. This is an approach and a set of measures developed by our colleague, Jill Gibbons from San Diego State University. And then as factors within the pathway of how these more upstream factors might impact prostate cancer outcomes, we're looking at a number of different social environmental factors, including crime, socioeconomic status, as well as built environment factors such as walkability and food environment. Okay. So within Project 1 of RESPOND, we actually have two different cohorts that we're looking at. The first is this cohort of the 10,000 men, African-American men with prostate cancer that we're recruiting across these different state-based cancer registries. But we have a second cohort that's unique to Project 1 that's just compiling the cancer registry data from these RESPOND regions. And so this is not based on survey data or recruitment, it's based on the existing data that already exists within these cancer registries. So we pulled it together, this massive data set of cancer registry data from California, Georgia, Louisiana, Detroit, New Jersey, and Texas. And with that, we were able to compile data on 124,000 African-American men and almost 512,000 non-Hispanic white men with prostate cancer. And so what I'm showing you here is for this cohort of African-American and white men, the distribution of the redlining index. Now, what this index tells us, so what this index does is Dr. Beyer took the Home Mortgage Lending Act data And she basically said, if we see in any particular neighborhood that is a census tract, if the odds, how does the rate of the denial of a home loan, an application for a home loan within that neighborhood compare to its surrounding area? If it's less than 1, that indicates less redlining in that immediate neighborhood relative to the surrounding area. If it's greater than 1, that means that the odds of a loan denial within that immediate neighborhood is greater than the surrounding area. So here, values less than 1, the blue indicates less redlining. Values greater than 1 indicates more redlining in the neighborhood that the men live in. And so what I'm showing you here are the neighborhoods, the values of redlining for the neighborhoods that the men live in at the time of diagnosis for non-Hispanic white and African-American men. And I'm also showing you the data, the distributions by states, as well as the for the entire Respond region. So what we see here first at a glance is that there's huge differences between non-Hispanic and African-American men. In terms of redlining, we see generally that non-Hispanic white men, the majority of them, they're much more likely to live in areas where there's less redlining compared to African-American men. And the second and that's consistent across all of the regions. And the second observation that we make is that there are differences across the region. So let's just focus on compare, for example, California, where we see that a little over 30% of African-American men live in areas where there's less redlining, 65% live in areas where there's more redlining. And then let's look at Detroit, where less than 10% of African-American men live in an area where there's no redlining, and greater than 90% live in an area where there's significant redlining. And in fact, the vast majority, over 50%, live in areas where the odds of loan denial in their neighborhood is more than 3 times greater than in the surrounding region. This is the second measure that was developed by Dr. Beyer using the same data source, the Home Mortgage Lending Act data. And this measure looks at the racial bias in mortgage lending. What this measure does is that it takes the home mortgage lending data and looks within a particular neighborhood, what's the odds of a denial of a home loan, of an application for a home loan for a Black versus a white applicant. And this is holding amount of the home loan constant. So what we see here is that the first thing that struck me, and I felt like I had to go back and did we calculate this correctly? Is so again here, if a value is less than 1, that means that the odds of denial of a home, that African Americans were less likely to be denied a home loan compared to white applicants in that particular neighborhood. If the value is greater than 1, that means that an African American applicant is more likely to be denied a home loan compared to a white applicant. Now, what do we see here? Across the board, consistently across the region, the vast majority of areas have values greater than 1. And again, we see differences comparing non-Hispanic to African-American men. And as we would, this intuitively makes sense, African-American men are more likely to live in areas where, compared to white men, are more likely to live in areas where there's less loan denial for their racial group. But not that much. The vast majority live in areas where there's more loan denial, where is racial bias in mortgage lending. And again, we see differences across the regions. And so does this matter? Does this even impact prostate cancer outcomes? So this is hot off the press analysis that we are starting to do. And so what we've done, because this is cancer registry data, we have really rich information such as mortality information through linkage to different vital statistics data sets. And what we've done is we ran models where we've looked at the associations between, here I'm just showing redlining, this redlining index, compared to overall mortality due to prostate cancer. This is just showing, these are just models specifically among African Americans with prostate cancer. So let me just kind of orient you to this graph. let's just focus on this first panel here, which is the California data. So this is a model, just California data. The solid line here at 1 is the reference group where there's no redlining. So compared to men who live in neighborhoods where there's no redlining, these are the hazard ratios or the relative rate of death due to prostate cancer associated with each increasing level of redlining. And so in California, we see that as we increase in values of redlining, we see an increasing risk of prostate cancer mortality. If we look at the highest level of redlining, 3.0, the risk of mortality is almost 50% higher compared to men, African-American men who live in areas with no redlining. Now, what's interesting here, and we're trying to understand this a little bit better and how to best model this, is that we see we definitely see differences across the different response regions, but there are also states in which we do see consistent patterns. For example, for Louisiana, we see fairly similar magnitudes of associations as we do see in California and New Jersey as well. But in Georgia and Detroit, and Texas is sort of in the middle, but certainly in Georgia and Detroit, there seems to be not as strong of an association. We're now trying to consider maybe, but we also know that there's significant amounts of segregation in Georgia and Detroit. So we're trying to understand perhaps the rate of mortality among those in the no redlining area is already very, very high. So this is work in progress. And with that, I want to just include a few final thoughts on where do we go from here? This is thoughts that have been percolating in my brain as I've been doing this kind of work and have the opportunity to be funded on projects where we can really leverage our epi training to be able to really see how the data play out within these health equity models. So I've been kind of compiling these thoughts over time. I think it's important, and certainly I've come to appreciate that we need to recognize that health inequities arise from fundamental causes. So thinking back to that cells to society model, the boxes at the very top, the fundamental causes, and that those drive the neighborhood context in which we live, play, work, and worship. And so therefore, when we do studies, it's important to examine what are the driving structural and institutional factors. More often than not, in fact, very much so more often than not, we're focusing on those factors towards the bottom of that multi-level framework. We need to start moving up that framework. And in some ways, it's actually easy to do this. And you could see from the studies that I showed you in lung cancer among Asian American female never smokers and among the prostate cancer among African American men, that if we have address data, just collect the address data, and having that piece of geographic information can really just open up the vast possibilities to be able to integrate all this rich geospatial data that exists out there to characterize our structural and neighborhood context. And because we know cancer and cancer outcomes aren't just a one-time thing, that there is not just a narrow window in which the biologic process occurs, and we also appreciate the impact of life course and early life exposures on cancer etiology and outcomes. And so therefore, we need to start to, and certainly studies are doing this, but it's a little bit more challenging looking at structural and neighborhood context, but it's important to measure this context over the life course. And we need models that help to guide us of how to assess cumulative effects and weathering. And the work that we're doing in RESPOND and the FAN study is an initial attempt at trying to connect all of this together, the structural context and neighborhood context and its impacts on biology. I think speaking as an epidemiologist, we have a lot of data needs. I think a lot of this we are sort of learning as we go. What are the best ways to operationalize, conceptualize and operationalize, and what data are available out there to measure structural context and social determinants of health? It's incredibly frustrating that there are no standardized measures. In the response study, we try to use as much as we can with very minimal modifications, measures that are already existing out there. We recognize that Respond at the conclusion of our study is going to be an invaluable resource. I mean, 10,000 African-American men with prostate cancer from a population-based study. So we really wanted to be able to have data that can be comparable to data that were measured, collected from large behavioral risk factor surveys or from other large cohort studies. I think we need to really collect data at as granular of a level as possible. The research that we've been doing on Asian Americans is a great illustration of this. The initial study based on EHR data, a lot of healthcare systems we were not able to include their data in that initial EHR-based study because they weren't collecting data on detailed Asian American ethnicity. Without that data, we have no way of characterizing what the disparities are. But we also recognize it can be tricky to collect these data, especially within a busy healthcare system and with lots of sensitivities out there about how these data will be used, profiling. So I think we need to partner with our healthcare professionals and healthcare systems administrators and leadership to develop and conduct training for and help to provide guidance for how to best collect this information within a clinical context. And finally, we need to assess the effects of intersectionality. It's not the way that we run or trained to run our epi models. We, look at the independent effects of each factor within a model one at a time, holding everything else constant. Well, that's not how real life occurs. Real life occurs because I am a female, I am a person of color, I am a first-gen immigrant. I lived in a neighborhood where I was one of the few Asians within the entire neighborhood. And what impact does that have on my health outcomes? And so it's important to examine within the research that we do, be sure we're collecting data on all of these factors, the interactions among multiple marginalized social statuses. And finally, bringing us back to an equity or social justice lens, to achieve health equity, we need to address the upstream social and institutional inequities within our societies. And so really ensuring that as scientists and researchers that we are doing the research to understand what are the impacts and how do we intervene upon these upstream factors, including social inequities by class, race, ethnicity, gender, sexual orientation, as well as institutional inequities, including the policies and laws and regulations. And that was all I had. Thank you so much.
Great. Thank you, Scarlett, very much. There's a couple of questions in the chat, and I would like everybody to add more. Let me just take a couple of them going in order. So Robert Weinberg, about your lung cancer studies, is asking about stratifying your analyses around subtypes. And I think this is getting to the heterogeneity of both the phenotype, the tumor phenotypes, but also the etiology that may underlie these diseases, and how do you consider that? Can you consider that? Thoughts about that?
Yeah, absolutely. Sorry I didn't present that. We had actually probably about 50 additional tables from that study that we had generated. And this is actually in our published paper in CEBP, and I'm happy to put the link into that paper later on. Fortunately, subtype information, histologic subtype information is very high quality in the cancer registry data that we linked to these two EHR data systems to. And so we definitely were able to look at what's the risk of lung cancer by smoking status, or what's the incidence rate of lung cancer, rather, I should say, by smoking status for adenocarcinoma versus different types of non-small cell subtypes versus by small cell. The numbers start to get fairly small, as you can certainly imagine. Adeno was really the most, was the largest group, which is understandable given that these are populations of never smokers. And so we definitely see differences by subtype. Again, the numbers start to get fairly small. So that's, stay tuned, those data will be reported in our papers.
Thanks. And maybe just as a brief follow-up on that, you talked about intersectionality, and I'm just thinking about the concept of intersectionality and the concept of figuring out, the group of people or subgroups that are of greatest interest for particular etiology or intervention is great. But now, how do you do that when you have 17 different groups plus tumor subtypes plus, and you end up with, lots of zero cells? And are there, I mean, the concept is great. It's sort of the implementation I'm thinking about. Are there novel methods? Can we move away from p-values and sample size, or how do you think about that?
I think we need better tools. I think that's a great question, Tim. I think we need the minds to develop the tools to be able to do that. I think right now well, for one, I will say that I think qualitative approaches are a great way to assess intersectionality. And there certainly are some really interesting and superb studies that have been done. And I've come to appreciate, I have no formal training in qualitative data analysis, but I've sort of had to do it and have come to appreciate just the richness and the importance of and the value that qualitative data can provide. So I think we absolutely should be looking at qualitative data as a way to assess that. But from a quantitative standpoint, you're absolutely right. We're talking about interactions, and you start to get very sparse very quickly. And do we always want to be relying on statistical significance to be able to say, well, yes, here's an interaction, and this, therefore, we are seeing an intersectionality effect. So I think we need better methods. That's the way we've been doing it, but we also recognize that it's completely inadequate. I will also add that I think in the response study was really an interesting concept in that the first time we went in with our grants reviewers, and I hope I'm not calling out any reviewers who may be on here, but had the good, I think, suggestion or pushed us to include not only African Americans, but also non-Hispanic white patients in our assessments. We push back on that because we thought, really we know there's a disparity, but really from a critical race theory lens, there are factors unique to the African-American population of African-American men that is impacting their disparity. And we need to really drill down and focus our resources that NCI we want you to give us to focus on what that unique set of factors and experiences are. And so I think it's not exactly getting at intersectionality, but at least from a limited resource standpoint, using that lens and perhaps limiting your population assessment might be one way to do that. Of course, you're not directly addressing the disparity, but we're certainly able to start to, rather than saying, oh, now we only have resources to recruit only 5,000 African-American men because we also need to recruit 5,000 non-Hispanic white men. We're able to spread that across the African-American sample and hopefully increase our power to look at some of these interactions.
Great. Yeah, thanks. A couple more questions. Shang Jianpan is asking about the how you determine PM 2.5 exposure history. And
yeah, I am not going to be able to give a satisfactory answer on this one at all, because this is a great area. This is a great example in which we collaborate with just really stellar environmental science colleagues Dr. Peggy Reynolds is my colleague and co-I on this grant, does a lot of the really excellent work. We have actually really rich resources measuring air pollution based on a combination of air pollution monitors that are placed throughout the state, and I'm sure throughout the nation, as well as lots of different data resources. And so there's a lot of creating types of approaches from GIS that are used to derive these very localized measures.
And I'm not sure if the questioner is coming from the Harvard system, but we have a very extensive group of people with a lot of experience in assessing those kinds of measures. And so if you're interested in learning more, we can certainly put you in touch with the people.
That'd be great.
Who do that. And
yeah,
there's
absolutely.
a lot of opportunity for that. So Seth Alper is asking about the data that you presented in prostate cancer cases, I think, and he's just wondering, how does that compare with the rates in controls, the population? So you talked about rates of redlining. Do you do any data whether there's any difference between?
I see. So how does the distribution of redlining look just in the general population, not among a cohort of prostate?
Are they different? Are the prostate cancers fundamentally different in some way?
Yeah, I took those slides out. And so I had those. And the reason I took those slides out is because, as you could see, just even looking at one single slide was a lot of data, but also because it was very similar. Mm-hmm. So we surprisingly did not see too much variability comparing at the prostate cancer cohort with a general distribution of redlining.
So can you from that imply that there is the redlining is not associated with etiology, but it's associated with outcome, or is that too much of a leap to make that inference? You may not be able to.
Yeah.
There's no.
Well, we didn't look at that's
maybe too much of a leap to.
That's a really interesting.
Yeah.
Yeah, I think that should be our next study.
Okay.
Because I think we can calculate rates, right? We can. Yeah.
So I mean, it's one of those questions. I think it goes to when all the data that you're describing is really interesting, but how do you intervene? Where do you, at what level do you intervene? Do you do policy? Do you change, do you do molecular therapeutics, whatever? But also, if the contextual variables are not part of etiology, but they're part of progression, then that might direct how you would intervene or where you might want to intervene based on where the effects are.
Exactly.
So,
yeah.
Good.
Yep.
So there's a question about the health insurance, and so I think that some of the issues that you showed may reflect population differences in other factors that you may or may not have measured. So can you comment on any of that?
Yeah. I'd have to look at health insurance. I'm pretty sure that was a variable that we pulled. So we've been having, just yesterday, in fact, our team had a really good discussion about this. Kirsten Beyer just recently published a paper in which she used a very similar kind of approach, where she looked at her measures of redlining and racial bias in mortgage lending linked to breast cancer mortality in Georgia. And her paper, she started with a DAG, and based on that, concluded that it was inappropriate to adjust for things like health insurance, treatment, other neighborhood characteristics, because they were in the causal pathway between these structural racism measures, redlining and racial bias in mortgage lending, and mortality. So we're sort of chewing on that in terms of how that, and so that's why the model I presented was very, very minimally adjusted, because we need to think more about, do we do causal mediation analysis? What are we really interested in, total versus direct, indirect effects? So that's kind of an ongoing. So I kind of kept it fairly basic, but we do have health insurance data from the different states. We certainly know from a state-by-state basis, the implementation of the Affordable Care Act was different. And so those are some of and that's, we also looked at it on a state-by-state level as a start as well.
Yeah, perfect. And there's a lot of great data you can use for years and ask these questions.
Absolutely.
We only have a minute, but just there's two other questions about the registry and the linkages. So Deb Schrag is asking, what would you put into registries that would help better this kind of data? How do you modernize them? And Hari Ayer is asking about linkages. How do you, create better linkages? So any comments about the data sets that you use?
That's a whole talk. I could talk.
Yes, it is. Yeah.
So I will just say, in response to Hari's comment, that is a really great comment. Our initial EHR-based study was delayed by probably 2 years because of needing to come up with legal agreements, both on the registry side and the healthcare institution side. So I will just leave it at that. That certainly is an area I think we could really work on. We, not meaning me, because I think it's the lawyers or whomever that need to really work on that. With regards to Deb's question, I think that, as you probably know, Deb and others, that SEER, the NCI program, is really moving towards modernizing cancer registry data through linkages, through machine learning and NLP approaches. And I absolutely think that's the direction that we should be working across. I think that the approach we're taking with Respond and with the EHR linkage, we have another study that involves 5 different registries, which also is going on 1.5 years now, just getting all the agreements in place. That it's not as easy as it sounds. You sort of feel like all the data are already out there. There's a key to link them all. Let's just do it. The legal processes and just are really just onerous and daunting. But if that can be streamlined, that's a super efficient and great way to modernize data with, without de novo data collection. Are there any data elements that can be sunsetted? I think we need to, the other area in which I think we should be collecting more data and looking at better ways to collect the data. I mentioned EGFR and ALK mutation status. We need more biomarker data, and we need to figure out better ways to get that into these population-based systems. Data that can be sunsetted, I hate sunsetting data. We don't need to, but I can also, as a cancer registry director, appreciate that you can't just keep adding on more and more data and asking people to collect more and more data. Even if a lot of it is automated, there's people on the other end doing all the QC and qualitative control, and there's cost and resources associated with that. But we are always looking at what variables are really just not good quality, what can we improve, what really is just kind of not really important anymore, and to take that off the list of,
So great, thank you. We're over time, so we'll have to stop. I wish we had more time to discuss, because it's really interesting, but I appreciate everybody who attended, and thank you so much, Scarlett, for a fascinating hour, and we'll appreciate everybody's feedback on this session. Thank you.
Thank you all.